You've found Dreamtime Science, part classroom, part lullaby, entirely devoted to satisfying your curiosity while you find your way to sleep. You know what's fascinating? I've been thinking about how life manages to not just survive, but actually thrive in places that seem completely inhospitable, like scorching hot springs or the darkest depths of the ocean. There's something remarkable about these tiny creatures and plants that adapt in ways we're only beginning to understand, and I can't wait to share some of their incredible stories with you tonight. If these gentle explorations bring you comfort, a quiet like or subscription would mean the world to me. Your support helps keep these peaceful journeys flowing. Before we begin, create your sanctuary. Nestle into your chosen spot, let your muscles soften, and allow your breath to find its natural rhythm. The night air is still, and somewhere in the quiet corners of your mind, the old persistent question stirs once more. How tenacious is life, truly? What is it about living things that compels them not only to endure, but to flourish in places where only the boldest imagination once dared to set foot? When the world outside is subsumed by shadows and the bustle of the day has faded, these questions seem to hum louder, as if seeking an answer older than memory and vaster than the arc of any single story. The human imagination has always circled around this question, drawn like moths to a distant flickering lamp. For centuries, our myths and stories have conjured creatures that shaped themselves to the harshest of lands. The salamander, said to bathe in fire, the frost giants brooding in eternal winter, the sea serpents coiled in lightless ocean trenches. And in the modern age, as our gaze stretched beyond our own blue planet, science fiction seized the mantle, offering up a parade of beings forged in the crucibles of otherworldly extremes. It is here, in the liminal space between possibility and invention, that the concept of the extremophile first began to germinate in the public mind, not merely as a biological curiosity, but as a symbol of how life, when pressed against the boundaries of the conceivable, can unfurl in ways that confound expectation. Close your eyes, if you wish, and let the narrative drift to the year 1960, when the bathyscaphe Trieste plunged into the Challenger Deep, the lowest known point in the world's oceans. There, in the utter blackness and crushing pressure, 8 tons per square inch, enough to collapse a submarine like a tin can, tiny shrimp-like creatures were spotted darting through the beam of a searchlight. For the first time, the world was forced to reckon with the possibility that life could not only survive, but thrive, in realms where even our machines strained to function. But the seeds for this revelation had long been sown in the collective consciousness. In early science fiction, alien life was often a cipher for the unknown. Its biology was shaped not by the requirements of narrative plausibility, but by the fevered dreamscapes of writers who looked up at the stars and imagined the unimaginable. Life on Venus swam in scalding, acid-tinged swamps. Martian warlords stalked barren deserts, their blood an exotic chemical, their organs tuned to thin air and bitter cold. In the pages of pulp magazines, life was not a fragile flame, easily snuffed, but a wild and mutable force, as stubborn as it was strange. Yet, as the decades unfurled and our understanding of biology deepened, a curious convergence began to occur. The stories spun from whole cloth by speculative minds started to brush up against the edges of scientific reality. The more we looked, the more we found real creatures that seemed to live out these science fiction fables on Earth itself. Take, for instance, the tardigrade, popularly known as the water bear, a creature so minuscule that 1,000 could dance on the head of a pin. For most of human history, such beings were invisible, their feats unimagined. Yet these tiny animals, first described in the late 18th century, can survive where nothing else can. The vacuum of space, boiling water, freezing radiation, lethal doses of X-rays. They dry themselves to husks, suspending life until fortune smiles again. and then, with the faintest suggestion of water, spring back into motion as if awakening from a long, untroubled sleep. The tardigrade's exploits have become legend in popular culture, appearing in television, literature, and even children's cartoons as a symbol of indomitable resilience. They are cast as the superheroes of the microbial world, avatars of possibility, their DNA a talisman against oblivion. But they are not alone in this gallery of real-world marvels. Consider the acid-loving archaea that dwell in the boiling pools of Yellowstone, their cells aglow with the pigments that shield them from lethal heat and acid, or the bacteria that feast on radioactive waste, quietly metabolizing uranium in the darkness beneath old reactors, or the worms that burrow into Antarctic ice, eking out an existence in water that never rises above freezing. Each is a stanza in life's rebellious verse, a testament to the adaptability that science fiction once reserved for the denizens of distant, hostile worlds. It is no accident that these discoveries have so captivated the public imagination. For in each there is a kind of narrative reversal, a migration of impossibility from fiction into fact. What once seemed the province of dreams and tall tales has been revealed through the patient work of science to be woven into the very fabric of our planet. Even as the scientific language of extremophiles became more precise, popular culture continued to riff on the theme, layering new associations and anxieties atop the bare facts. In the 1982 film The Thing, a shape-shifting alien thaws from Antarctic ice, its biology so mutable that it can mimic any life form it encounters. The horror lies not only in its brutality, but in its radical adaptability. It is the ultimate extremophile, unbound by the limitations of terrestrial life. Later, in Star Trek and Doctor Who, entire civilizations are imagined to have arisen in cauldrons of lava or beneath the crushing atmospheres of gas giants, or in the methane seas of Titan. These stories borrow freely from the logic of the extremophile, that life, given time and the barest foothold, will find a way to bend even the harshest environment to its will. The fascination has a deeper psychological root, perhaps. The extremophile is, in some sense, a mirror for humanity's own aspirations and anxieties. We are a species forever pressing against our own limits, crossing deserts, scaling mountains, plunging into the abyssal deeps, launching ourselves into the void of space. The creatures that thrive where we would perish offer both reassurance and a challenge. They whisper that the universe is not so hostile as we once feared, that life is more versatile, more robust than our ancestors dared hope. But they also remind us that we are not the measure of all things, that the forms and strategies of existence are more various and surprising than any single imagination can encompass. In the early 21st century, this motif has found new expression in the search for life beyond Earth. The icy moons of Jupiter and Saturn, Europa and Enceladus, are now seen not as barren wastelands, but as possible havens for extremophiles. Under their frozen crusts, vast oceans may roil in darkness, heated from below by tidal forces and radioactive decay. Here, perhaps, are analogues to the heat-loving bacteria of Earth's undersea vents, living out their strange, slow lives in utter secrecy. Science fiction has leapt eagerly into this breach. Novels and films speculate on the forms such life might take. Blind, pallid fish with phosphorescent organs, microbial mats that draw energy from volcanic seeps, creatures that communicate through pulses of electricity or chemical gradients, untethered from the familiar senses of sight and sound. These imaginings are not arbitrary, but grounded in the logic of extremophiles, extrapolations from what we know to what we hope or fear might be. Yet even as the idea of life's adaptability has filtered into the mainstream, it retains an aura of the uncanny. There is something unsettling about organisms that flourish in acid, or drink radiation, or bask in boiling oil. They seem to violate an unspoken order, to invert the expectations that comfort us. If life can survive anywhere, what does it mean to call a place uninhabitable? Does our own definition of habitability serve us, or does it merely reflect the narrow band of conditions to which we ourselves are suited? These questions echo through the stories we tell, blending science and myth, fact and fiction. The extremophile is both a symbol and a challenge, a prod to the imagination, a prompt to humility. It invites us to reconsider the boundaries of our own experience, and to wonder what other forms might lurk just beyond the reach of our current understanding. From the hot springs of Kamchatka to the sulphurous plumes of Io, from the radioactive ruins of Chernobyl to the frozen lakes of Antarctica, the real and the imagined have begun to merge. What was once the domain of speculative fiction is now the terrain of explorers and microbiologists, their instruments tuned to the faintest pulse of life in places where, until recently, no one thought to look. In the weave of popular culture, extremophiles have become shorthand for the unexpected, the resilient, the alien within the familiar. They populate the margins of our stories, both as monsters and as marvels. Sometimes they are threats to be contained, like the mutating viruses of apocalyptic thrillers or the parasitic invaders of cosmic horror. At other times, they are emblems of hope, the seed of life that might survive catastrophe, or the messenger from another world that opens the door to cosmic kinship. There is a subtle irony in all this, for even as we project our fears and aspirations onto these creatures, their true story is one of quiet, patient persistence. Most extremophiles do not conquer their surroundings in any dramatic sense. They adapt, they endure, they refine the machinery of their existence until what once seemed impossible becomes, for them, routine. Their genius is not in defiance, but in accommodation, an intricate dance with entropy and adversity that has played out for billions of years. The science fiction of extremophiles, then, is less a tale of conquest than of transformation. It asks us to imagine not only new forms of life, but new ways of being alive, to envision existence as a spectrum, broad and variegated, rather than as a ladder with humanity at the pinnacle. In this way, the old stories of salamanders in fire or giants in ice begin to resonate anew, reframed by the discoveries of modern science. As you drift on the edge of sleep, consider the possibility that the universe is not a hostile void, but a vast, intricate landscape of opportunity, one in which life, in its myriad forms, is forever probing, forever experimenting, forever rewriting the rules. The next time you see a patch of moss clinging to a city wall or a bird nesting in a crumbling ruin, remember the legacy of the extremophile, that the improbable is only improbable until it is observed, and that the boundary between fiction and reality is as thin and shifting as the membrane of a tardigrade cell. Somewhere in the darkness beneath a frozen lake, or in the cauldron of a volcanic vent, another stanza of life's rebellious verse is being composed. Its rhythm is slow, its language unfamiliar, but its meaning is clear. Life will not be confined to the narrow corridors of comfort. It will leap the barricades, infiltrate the forbidden, and claim as its own the places we once thought insurmountable. And so the question lingers, unresolved, like the last chord of a half-finished song. If life can flourish in acid and vacuum, in boiling oil and freezing brine, what other forms might it take, out there, beyond the reach of our current dreams? What new stories are waiting to be told in the unlit corners of possibility? Beneath the gentle hush of twilight, the world's pulse slows, and we find ourselves wandering along the rim of possibility, a place where life, in all its unyielding tenacity, wrestles with the very boundaries of existence. The tale of survival is not merely one of brute resilience or heroic adaptation, but an intricate, ongoing serenade, each organism improvising its own melody against the backdrop of harshness, uncertainty, and change. Let us journey now into the heart of this serenade, to witness the subtleties and marvels of survival beneath the grand surface. The limits of life are not simply drawn by temperature, pressure, or dryness, but by the intricate worlds of biochemistry and physiology, each a silent negotiation with the world's extremes. In the brooding depths of the Marianas Trench, where sunlight dies and the water is pressed with the weight of 1,000 atmospheres, a peculiar kind of life flourishes. The Hadal snailfish, ghostly and translucent, drifts over the silt. Its body is not armoured against the crushing dark, but rather softened. Its bones are more cartilage than calcium, its flesh almost gelatinous. High pressure would collapse proteins and cell membranes in most creatures, but the snailfish and its kin have evolved to fill their cells with stabilizing molecules, osmolytes, like trimethylamine N-oxide, TMAO, which counteract the destabilizing force of pressure. Here we see adaptation not as a rigid defense, but as a supple yielding to the environment's demands, a chemical embrace of adversity. Yet, the myth persists that adaptation is a kind of magical, all-encompassing shield, that given time, life can conquer any setting, no matter how forbidding. This is a comforting illusion. In truth, the boundaries of survival are precise and unforgiving, defined by the thermodynamics of molecules and the fragile balance of living membranes. Take the story of the heat-loving archaea, those denizens of scalding hot springs and submarine vents, whose enzymes and cell walls remain stable at temperatures that would unravel the proteins of most other life. Their secret lies in the architecture of their molecules, a greater proportion of heat-stabilizing amino acids, more rigid ring structures in their cell membranes, and protective chaperone proteins that shepherd other molecules through the fevered chaos. But even these adaptations are not infinite. Above a certain threshold, the very chemistry of life begins to fail. DNA unwinds, enzymes falter, and the machinery of cells grinds to a halt. The upper thermal limit for known life hovers around 122 degrees Celsius, with the microbe Methanopyrus candleri holding the record. Beyond this, life as we know it cannot persist. The converse is equally true. In the frozen soils of Antarctica and within the permafrost of Siberia, bacteria and nematodes enter a suspended animation, their biochemistry slowed to the barest trickle. Some produce antifreeze proteins that bind to ice crystals, halting their growth and preventing the lethal rupture of cell membranes. Others accumulate sugars like trehalose, which glassify the cell's contents, protecting the delicate machinery from damage during freezing and thawing. But there is always a lower bound, a temperature at which molecular motion ceases altogether and the dance of life is arrested in the silence of absolute zero. No adaptation can cheat the fundamental laws of physics. We must be careful, then, not to conflate adaptability with invincibility. The evolutionary process is a slow, meandering river, not a magic wand. Adaptations are trade-offs, each one a compromise with costs as well as benefits. For instance, the camel's ability to retain water and survive in the parched Sahara comes at the expense of speed and agility. Its lumbering gait is the price it pays for a physiology built around water conservation. The Antarctic icefish, whose blood is clear and nearly devoid of hemoglobin, survives in oxygen-rich freezing waters thanks to antifreeze proteins, but would suffocate in warmer, less oxygenated seas. The very traits that enable survival in one context can become fatal liabilities in another. And so we observe, throughout the living world, a mosaic of solutions, each one elegant, but none omnipotent. The tardigrade, often mythologized as the indestructible water bear, seems at first glance to flout every limit. subject it to boiling, freezing, radiation, or the vacuum of space, and it emerges, eventually, from its cryptobiotic slumber, shaking off damage that would obliterate most other life forms. The tardigrade's secret lies in an uncanny ability to desiccate its body almost completely, entering a state called a ton, where metabolic activity nearly ceases, and special proteins, called intrinsically disordered proteins, form a glassy matrix around cell structures. DNA repair mechanisms spring into action upon rehydration, patching up the ravages of radiation or oxidation. Still, even this champion is not truly immortal. Repeated cycles of stress or conditions outside the narrow window of recovery will eventually claim its life. The legend of perfect adaptability dissolves under the scrutiny of detail. Woven into this tapestry are the subtle complexities of biochemistry, often invisible to the naked eye. The ability of halophiles, salt-loving microbes, to thrive in brine pools and salt pans relies on a delicate balancing act, adjusting their intracellular salt concentrations to match their surroundings. Normally, high salt would denature proteins, but these organisms have evolved proteins with surfaces studded with acidic amino acids, rendering them stable and functional in saline onslaughts. Yet, remove them from their briny homes and they perish quickly. The very adaptations that protect them become their undoing elsewhere. Consider also the deep, silent caves of our planet, where sunlight has never penetrated, and the air is heavy with the mineral scent of stone. Here, blind cavefish pick their way through the blackness, their eyes vestigial, their pigmentation faded into ghostly pallor. Deprived of sight, they have honed other senses, taste buds and lateral lines, sensitive to the faintest vibrations and chemical traces in the water. Evolution, in these depths, is not about adding ever more features, but about paring away the unnecessary, refining the essential. Energy, always at a premium, is not wasted on vision when darkness reigns eternal. Adaptation, thus, is a sculptor, carving away as much as it builds. The intricacies of adaptation are not limited to the extremes of temperature, pressure, or darkness. In the high Andes, where oxygen is scarce and the air thin, the bodies of people and animals have responded across generations. The hemoglobin in their blood binds oxygen more tightly, and capillary networks permeate their tissues more densely. Yet, even here, there are limits. Altitude sickness besets new arrivals, and above certain elevations, even the hardiest cannot long survive. The Tibetan yak, with its oversized lungs and efficient metabolism, thrives where other cattle would falter, but bring it down to lush, lowland pastures, and it finds itself out of breath, a high-altitude specialist unsuited for the abundance below. At the cellular level, these adaptations are orchestrated by an array of molecular switches, genes that turn on and off in response to environmental cues. Epigenetic changes, subtle chemical modifications to DNA and histones, shape which genes are active, allowing organisms to fine-tune their physiology without altering the underlying genetic code. These changes can sometimes be passed on to offspring, blurring the line between hardwired evolution and flexible, real-time adjustment. Yet this flexibility too has its boundaries. There are forms of stress that no amount of genetic or epigenetic reshuffling can mitigate, where the environment simply exceeds the toolkit of adaptation. One persistent myth, seductive in its simplicity, is that all life is perpetually striving toward greater complexity and fitness. In reality, many adaptations are not about progress, but about sufficiency, a good enough solution that works under current conditions. Life's history is littered with experiments abandoned, with forms exquisitely adapted to circumstances that no longer exist. The horseshoe crab, virtually unchanged for hundreds of millions of years, is not striving for something better. It is simply well matched to its environment. When that environment changes, as it unavoidably does, adaptation may falter, and extinction becomes the coda. The limits of adaptation are also written in the language of biochemistry, where the rules of chemistry and physics set unyielding boundaries. Enzymes, those remarkable molecular machines, operate most efficiently within a narrow band of temperature, pH, and salinity. Push them too far, and their three-dimensional shapes unravel, their catalytic prowess lost. The lipid bilayers that form cell membranes must remain fluid enough to permit movement, but not so fluid as to become leaky. The very molecules that make life possible are themselves fragile, their stability hemmed in on all sides by the environment's demands. Yet, within these constraints, life improvises endlessly. The extremophilic Archaeon Picrophilus oshimai can grow at a pH of 0.06, more acidic than battery acid, thanks to a cell membrane that is virtually impermeable to protons. In alkaline lakes, other microbes survive by maintaining an interior several orders of magnitude more acidic than their surroundings, spending precious energy to pump ions in and out. The cost of survival is never zero. Every adaptation exacts a price, whether in energy, speed, reproductive success, or vulnerability to other stresses. Humans, too, are bound by these limits, though we often imagine ourselves uniquely capable of transcending them. Our bodies are finely tuned to the planet's surface conditions, temperatures, pressures, oxygen levels, and radiation. In the vacuum of space, or the crushing darkness of the deep sea, we rely on technology to extend our reach. Our adaptations are not genetic, but cultural and technological, our survival strategies encoded in blueprints and stories rather than DNA. The boundaries of human adaptation are defined not just by our biology, but by our ingenuity, our willingness to build shelters, don suits, and craft environments within environments. Still, there are mysteries that elude us, corners of adaptation that remain unsolved. How do certain seeds survive for thousands of years, only to awaken with a drop of water and a ray of sun? What are the hidden limits of life's endurance? Are there realms, perhaps beneath the surface of Mars or the icy moons of Jupiter, where biochemistry could take forms unimagined on Earth? The Serenade of survival is not finished. It is an ongoing composition, its notes echoing through the ages, always flirting with the edge of the possible. As we rest here, poised on the boundary between what is known and what is yet to be discovered, the complexities of adaptation unfold like a slow, enigmatic melody. Each solution is provisional, each boundary both a barrier and an invitation. The serenade continues, its themes weaving through the fabric of life, hinting at deeper harmonies yet to be revealed. And as night deepens, one cannot help but wonder at the next movement in this grand, unfinished symphony. Long before humans could even dream of exploring the most forbidding places on Earth, our ancestors gazed at bubbling springs and steaming vents with awe and trepidation. They saw in these places the breath of the gods, or the entrance to the underworld, never suspecting that life itself might thrive in such inhospitable realms. It would take centuries, centuries of invention, of boldness, and of quiet, persistent curiosity, before the invisible denizens of the extremes would come into view. Yet, even as the boundaries of the known world stretched outward, it was the slow evolution of our instruments, the tools of discovery, that would ultimately unravel the secrets of life at the edge. In the earliest days of biological exploration, the world was surveyed with little more than the naked eye. The discovery of microorganisms in the 17th century by Antonia van Leeuwenhoek, peering through his handcrafted lenses, was a revelation. Yet, for all their ingenuity, those first simple microscopes could never penetrate the darkness of the deep ocean, nor could they peer into the scalding heart of a hydrothermal vent or tease apart the delicate threads of DNA that encode life's resilience. The story of extreme life is, in many ways, the story of the instruments we have built to perceive it. The journey began on the surface, where the tools of classical biology, microscopes, petri dishes, glass pipettes, allowed us to survey the teeming world of microbes in soil and water. Yet, even as the field matured, there remained vast swathes of the earth where life was simply assumed to be impossible. The boiling acid pools of volcanic craters, the pitch-black trenches of the abyssal sea, the icy wastes of Antarctica. These were places beyond our reach and beyond the imagination of most scientists. But curiosity, like life, finds a way. The challenge of the extremes drew a rare breed of pioneers, engineers, geologists, biologists, and dreamers, all united by a desire to see what lay beyond the familiar. One of the first great leaps came with the invention of the deep-sea submersible. In 1960, the bathyscaphe Trieste, crewed by Jacques Piccard and Don Walsh, descended into the Challenger Deep, nearly 11 kilometers beneath the surface of the Pacific. As the tiny vessel creaked under the crushing weight, the explorers peered out through a thick plexiglass window into the eternal darkness. To their astonishment, they glimpsed a flatfish gliding across the sediment, a living creature in a realm thought utterly sterile. The presence of such life at the ocean's deepest point hinted at hidden ecologies, and it was the beginning of a new era in biological exploration. Yet, the real revelation was still to come. In 1977, a team aboard the research vessel Alvin ventured to the mid-ocean ridges near the Galapagos Islands, guided by faint chemical clues and the flickers of their instruments. What they found was beyond anything science had predicted. Hydrothermal vents, spewing superheated water rich in minerals, and clustered around them, lush gardens of life, giant tube worms, clams, crabs, and swarms of peculiar bacteria. Here, in the absence of sunlight, life flourished, drawing energy not from the sun, but from the chemistry of the earth itself. The discovery of chemosynthetic ecosystems shattered our assumptions about the limits of life, and it was made possible by extraordinary technical ingenuity. The robust hull of Alvin, her manipulator arms reaching into the vent fields, her cameras capturing the alien landscape. Exploring such places, however, required more than just physical presence. The temperatures near hydrothermal vents soar to over 350 degrees Celsius, while the pressure outside is hundreds of times greater than at sea level. To sample these environments, scientists engineered specialized devices, insulated samplers, titanium bottles, and remotely operated vehicles, ROVs, that could withstand the extremes. These mechanical emissaries brought back not only samples of water and rock, but also the living organisms themselves, many of which would wither instantly in the gentler conditions of the surface. To keep these creatures alive, researchers designed pressurized aquaria, small steel chambers that could simulate the crushing depths and searing temperatures of the vent fields. It was in these tiny ecosystems that scientists first isolated thermophilic bacteria, organisms that not only survived but thrived at temperatures that would kill most known life. Under the microscope, these bacteria appeared unremarkable, yet their biochemistry was a revelation. Their proteins were stable at temperatures that would denature ordinary enzymes. Their cell membranes were built from unusual lipids, impervious to heat and acid. Each adaptation was a clue, a fragment of the grand puzzle of life's possibility. As the tools grew more sophisticated, so too did our questions. It was no longer enough to simply see these organisms. We wanted to understand how they worked. Enter the molecular age, a revolution fueled by the ability to dissect life at the level of genes and proteins. The polymerase chain reaction, PCR, invented by Carrie Mullis in the 1980s, allowed scientists to amplify DNA from even the tiniest samples. Suddenly, the genetic blueprints of thermophiles, acidophiles, and halophiles could be read and compared, their evolutionary histories reconstructed. One of the most profound discoveries came from the study of a humble microbe, Thermus aquaticus, isolated from a boiling spring in Yellowstone National Park. Its heat-loving DNA polymerase became the engine of the PCR revolution, enabling the rapid amplification of genetic material and launching the field of modern molecular biology. This single enzyme, born in the cauldron of an extreme environment, now underpins everything from medical diagnostics to forensic science. It is a testament to the power of studying life at the edge. What seems exotic and rarefied may in time become indispensable to all. But the quest to understand extremophiles has not been a solitary endeavor. It is a tapestry woven from the work of countless pioneers, each pushing the boundaries of what is possible. Carl Woese, a microbiologist with a restless intellect, used the emerging tools of molecular phylogenetics to peer into the deep history of life. By comparing the sequences of ribosomal RNA, he discovered a new domain of life, the archaea, whose members were first found in extreme environments. This revelation redrew the tree of life itself, revealing that the familiar world of plants, animals, and fungi was but one branch amid a far vaster and stranger diversity. Elsewhere, scientists like Thomas Brock and Hudson Freeze braved the scalding geysers of Yellowstone, scooping up microbial mats and culturing them in makeshift laboratories. Their persistence paid off with the isolation of new species, each more astonishing than the last. In the salt-crusted ponds of the Dead Sea, researchers uncovered halophilic archaea, organisms whose cells teem with salt, their proteins exquisitely tuned to resist dehydration. In the acidic streams of Rio Tinto, bacteria painted the rocks with vivid hues, their metabolism fueled by iron and sulfur. To probe the limits of microbial endurance, some experiments have been staged in the cold vacuum of space itself. The European Space Agency's EXPOSE missions, for example, mounted samples of microbes, lichens, and spores on the exterior of the International Space Station, exposing them to solar radiation, extreme temperatures, and the relentless bombardment of cosmic rays. To the astonishment of many, some survived, their resilience challenging our assumptions not only about life on Earth, but about its potential elsewhere in the cosmos. Yet for all these triumphs, the study of extremophiles is as much an art as a science. The environments themselves are treacherous, access is fleeting, and the organisms are often exquisitely sensitive to change. To sample a hot spring without killing its inhabitants requires a blend of improvisation and care. To decipher the metabolism of a deep-sea archaeon demands patience and ingenuity. The tools have become ever more subtle. Microelectrodes that measure the faintest whiff of oxygen or sulfide, metagenomic sequences that reconstruct entire microbial communities from a handful of DNA fragments. The data they yield is vast, complex, and often ambiguous, a mosaic of clues to be assembled with imagination and rigor. There are moments of serendipity as well, those flashes of insight that arise from chance and boldness. In the icy lakes beneath Antarctica's glaciers, robotic probes have sampled ancient waters sealed off for millennia. To the delight of their creators, they have found not sterility, but thriving ecosystems, microbes eking out an existence on the barest trickle of chemical energy. In the caves of Romania, far from sunlight, entire communities have evolved that breathe sulfur and methane, their genes a living archive of adaptation. And always, the tools evolve. Where once we relied on what we could extract and culture, now we peer directly into the genetic code of entire environments. Metagenomics, the sequencing of all the DNA in a sample, has revealed a microbial universe beyond imagining, vast networks of genes, many with no known function, hinting at metabolisms and capabilities still undiscovered. The uncultured majority, once invisible to science, is now coming into view, an invisible biosphere woven into the fabric of every extreme place on Earth. Some of these discoveries are almost philosophical in their implication. That life can flourish in boiling acid, in the darkness of the deep sea, or beneath glaciers, suggests a universality of adaptation, a kind of stubborn protean creativity at the heart of biology. It invites us to wonder not just about the Earth, but about the cosmos. Are there analogues to our hydrothermal vents on the icy moons of Jupiter or Saturn? Could life, in some alien guise, be thriving in the methane lakes of Titan or beneath the frozen crust of Europa? The tools of discovery have now become instruments of speculation as well. Robotic landers, spectrometers, and orbiting telescopes are being designed to search for the chemical traces of life far beyond our planet. The lessons learned from extremophiles about biochemistry, about resilience, about the unexpected diversity of the possible inform every such quest. Yet, for all our technical sophistication, there remains something deeply human at the core of this endeavor, a sense of humility and of wonder. Each new tool, each clever experiment is an extension of our senses, a way to reach out into the darkness, to feel for the pulse of life in places we once thought empty. And with each success, the world grows a little larger, a little stranger, and infinitely more alive. As night deepens and the world outside falls silent, the story of our pursuit continues. In laboratories bathed in the blue glow of monitors and beneath the shifting ice of remote frontiers, new experiments unfold. The tools at our disposal are sharper than ever, but the mysteries remain, beckoning us onward, toward the next revelation, the next encounter at the boundary of the possible. The history of discovery is not a straight line, but a winding path, full of detours and surprises, each shaped by the tools we wield and the questions we dare to ask. And so the lens turns once more, this time to the future. What new instruments will we conjure? What extremes remain to be explored? The answers, as always, lie just beyond the horizon, where our curiosity meets the unknown. Night drapes its velvet curtain over the world, and the last embers of sunlight flicker out beyond the horizon. In this hush, it is easy to feel small, to sense the immensity of the universe pressing in at the edges of our awareness. Yet, within this stillness, a profound question arises, one that has echoed since the first humans looked up at the stars with wondering eyes. What is it that binds life, in all its dazzling variety, to this universe of extremes? What is the secret thread that allows living things, from ancient bacteria to our own restless species, to carve out existence against the odds, to adapt, endure, and sometimes even thrive in the harshest corners of creation? To ponder this is to step softly into the heart of mystery. For, as we have seen, life's resilience is not merely a catalogue of clever tricks. It is something deeper, a testament to the sheer inventiveness of biology and to the intimate conversation between living matter and the physical world. The extremophiles, the acid lovers, the heat dwellers, the creatures that swim in liquid methane or burrow beneath Antarctic ice, are not exceptions to the rule, but perhaps its most eloquent exemplars. They whisper to us that life is not a fragile flame, flickering only under gentle conditions, but a force capable of kindling its fire in the howling wind and the frozen dark. What does it mean, then, to exist at the edge, to inhabit the boiling vent, the sunless cave, the saline desert, or the radioactive ruins? The adaptations we have explored are not only strategies for survival. They are also acts of profound creativity, of living matter, writing poetry with the language of chemistry and physics. When a bacterium crafts a protein that folds flawlessly at 100 degrees Celsius, when a tardigrade knits itself into a glassy cryptobiont to survive the vacuum of space, it is performing a dance as ancient as the planet itself, a dance that we too have joined in our own way. For humanity, the story of adaptation to extremes is both literal and metaphorical. On the one hand, we are creatures of biology, shaped by the same evolutionary pressures that have honed the enzymes of hyperthermophiles and the membranes of acidophiles. On the other, we are storytellers and toolmakers, able to step beyond the limits of our own flesh by dreaming, building, and imagining. Our resilience is not measured only in genes, but in ideas. The fire we learn to master, the clothing fashioned against the cold, the language spun across the chasms of ignorance. The thread of adaptation runs through our hands as surely as it does through the flagella of archaea. To reflect on this is to blur the boundary between the scientific and the philosophical. Consider the notion of homeostasis, the ceaseless balancing act by which living things maintain their inner constancy amid chaos. The term evokes images of cells adjusting their salt and sugar, of lizards basking in the sun, of humans sweating beneath the noon sky. Yet on another level, it speaks to a universal yearning for equilibrium, for harmony amid uncertainty. Is it not, after all, what we seek in our own lives, a way to persist, to find meaning, to adapt to the storms and droughts that sweep through the landscape of experience? The extremophiles, in their silent wisdom, teach us that stability is not stasis. Life's greatest triumphs emerge not from hiding from adversity, but from embracing it, from using the harshness of the world as raw material for invention. The proteins that resist boiling, the DNA that shrugs off radiation, the metabolic pathways that flourish in poison, these are not mere shields against destruction, but keys that unlock new ways of being. In every adaptation, there is a risk, a gamble, a wager that the path less traveled may hold undiscovered treasures. For humanity, too, our own history is written in the ink of adaptation to extremes. We have wandered out of Africa into ice and desert, crossed oceans and mountains, survived plagues and famines, rebuilt from the ashes of disaster. Our resilience is not only physical, but cultural, a capacity to imagine futures different from our present, to dream of other worlds, and then set sail toward them. The same restless curiosity that led us to peer into the hydrothermal vents of the deep sea, or to drill into the frozen lakes of Antarctica, is the spark that ignites our greatest achievements, and sometimes our gravest follies. But what of the future? As we gaze into the uncharted tomorrows that lie before us, the lessons of the extremophiles take on new urgency. Our planet is changing. The climate warms, the seas rise, the forests burn and wither. New extremes beckon, some of our own making, others as old as the planet. The adaptability of life is both a reassurance and a warning. On the one hand, it whispers that life can persist, that even when the familiar vanishes, something may endure, learn, and flourish. On the other, it reminds us that adaptation has its price, that not all who face the storm will survive unchanged. Yet in this, there is also hope. For if life's great secret is its capacity for transformation, then we too are heirs to that legacy. We are not prisoners of our present, nor of our past. Our technologies, our cultures, our science, these are extensions of the same evolutionary impulse that carved the first ribosome or honed the first photosynthetic pigment. If the acid-loving archaea can retool their entire cell membranes for survival, if the tardigrade can rewrite the script of metabolism to weather cosmic emptiness, might we not also discover new ways to live with and within the extremes we now face? There is perhaps a deeper lesson here, one that glimmers beneath the surface of biology and physics, a lesson about humility and wonder. The universe, in all its grandeur and terror, is not arranged for our comfort. The boiling vents, the frozen wastes, the toxic lakes, they are not inhospitable so much as indifferent, shaped by laws and forces older than life itself. Yet against this backdrop, life has found a way to write itself into the story, to carve a niche, however slender, in the vastness of possibility. To contemplate this is to sense a kinship, a quiet fraternity, with all that lives and strives. The extremophiles are not merely curiosities in the margins of biology. They are our distant cousins, fellow travellers in the long journey from non-living chemistry to consciousness. Their story, like ours, is one of audacity, a refusal to accept the boundaries of the possible, a willingness to leap into the unknown. The cosmic dance of adaptation, played out in 1,000 forms, binds us to them and to the universe itself. And so, as humanity peers outward into the abyssal depths, the Martian deserts, the icy moons of Jupiter and Saturn, our search for life elsewhere is also a search for ourselves. Will we find, beneath the crust of Europa or in the haze of Titan, beings who have learned to thrive in alien extremes? If so, what will they teach us about the nature of life, about the ways that chemistry can give rise to memory, intention, and perhaps even meaning? If not, what does it say about the rarity, the preciousness of our own existence? There is a paradox at the heart of this quest. The more we learn of extremophiles, the more we see that the boundaries of life are wider than we ever dreamed. Yet each adaptation is also a thread that ties life to its particular world, its unique history. The acid-loving microbe is exquisitely suited to its sulphurous springs, but would perish in a freshwater lake. The deep-sea vent worm flourishes in darkness and pressure, but cannot survive the sunlit shallows. Adaptation, then, is both liberation and constraint, a way to transcend the ordinary, but also a tether to the particulars of place and time. For us, too, this duality is ever present. Our ability to adapt allows us to inhabit almost every corner of the earth, to bend nature to our will. Yet we remain vulnerable, dependent on the thin skin of atmosphere, the fragile cycles of water and carbon, the balance of sunlight and shade. Our resilience is real, but so is our fragility. Like the extremophiles, we are both conquerors and custodians of our world, heirs to its abundance and stewards of its future. This reflection, quietly unspooling in the stillness of night, leads us to consider the philosophical heart of adaptation, the search for meaning in the face of indifference, the forging of identity amid chaos. To adapt is not merely to survive, but to assert, however softly, that life matters, that it is worth persisting, worth transforming, worth celebrating. Each extremophile, in its improbable persistence, is a testament to the value of existence itself. Each human act of resilience, from rebuilding after disaster to dreaming of distant planets, is an echo of the same affirmation. Under the cold gaze of the cosmos, it is tempting to feel insignificant, to shrink before the scale of the universe and the harshness of its extremes. But the lesson of life's adaptation is otherwise. In every extremophile, in every act of survival, there is a quiet defiance, a refusal to be erased. Life does not merely endure, it creates, it experiments, it dares. The boiling vent, the acidic lake, the radioactive ruin, these are not merely obstacles, but canvases upon which the artistry of biology is painted. Looking forward, the implications for our future are as profound as they are unresolved. The adaptability of life suggests that, even as the world changes, new forms may arise, new possibilities may unfold. Yet it also challenges us to consider the limits of adaptation, biological, cultural, ethical. How far can we stretch before the thread snaps? What will it mean to adapt to a world we have altered beyond recognition? Can we learn from the extremophiles not only how to survive, but how to flourish, to find beauty and belonging even in the harshest conditions? In this, perhaps, lies the most important connection between the story of life's resilience and our own journey. The extremophiles are reminders that life is not a passive thing, waiting for the world to become gentle. It is an active force, a maker of futures, a sculptor of possibility. In their example, we find both caution and inspiration, a call to respect the limits of our world, to cherish its diversity, but also to imagine new ways of being, new forms of harmony between life and the universe. As you drift towards sleep, let these reflections settle gently in your mind. The world is vast and filled with extremes, but also with possibility. Life, in all its forms, is a testament to the power of adaptation, to the mystery of emergence, to the enduring quest for meaning. We are part of this story, shaped by it, challenged by it, called to contribute our own verses to the ongoing poem of existence. In the silent spaces between breaths, between thoughts, the universe waits, patient, indifferent, beautiful. Life, in its infinite adaptations, answers with creativity, with persistence, with hope. The dance continues, unfinished, unresolved, ever unfolding. And in that unfolding, we find our place, our purpose, our profound connection to all that is and all that might yet be. As our journey through the resilient whispers of life in the universe's most hidden corners gently drifts to a close, may you carry the quiet wonder of these extraordinary adaptations like a comet tail fading into the hush of space. Rest easy beneath the cosmic quilt, and when your heart yearns for more Dreamtime tales, return to wander once again among the stars and secrets of our vast, mysterious cosmos.